So good morning, my name is Glenn Thorlby and I'm a research leader at Scion in the area of plant biotechnology. And today I'm going to tell you a little bit about several technologies or biotech-based technologies which we believe that over the next few years will add real value to conifer breeding programs. And now to start the presentation. So today, as I just said, I'm going to talk about the transformational opportunities that several new breeding technologies offer to conifer breeding programs. During the presentation, I will give some examples of how the technologies described are being used in the MB Winning Against Wildings program. For those of you who are not familiar with this, it is a multidisciplinary project looking at all aspects of the wilding problem in New Zealand, from ecology through to providing solutions to existing and future wildings. Some of the work in this programme, particularly work focusing on producing low coning or sterile Douglas-fir, is supported by the forest growers. The key aim of the project is shown here, which is to use biotechnology to stop essential genes or processes that are required for coning, and thus prevent new wildings. This will enable future plantings of Douglas-fir, which, although it is prone to wildings, is highly productive in parts of the South Island. I'm going to describe 4 new breeding technologies that are either starting to be used or already in use in agricultural plant breeding programs, and also provide exciting opportunities for forestry. The first 2 technologies, transgenic modification and gene editing, alter the DNA sequence. In the case of transgenics, this has led to organisms produced using it being classified as GMOs, whilst gene editing, because of the nature of the changes made, is classed in much of the world as a non-GMO. Currently, this is not the case in New Zealand, but I think over the fullness of time, New Zealand will align with global regulations. The other 2 technologies, exogenous RNAi and epigenetic modification, do not alter the DNA sequence itself, but they alter the way that genes work, and because of this, are globally considered to be non-GMO. Transgenic technology is probably what comes to mind when you think of genetically modified organisms or GMOs. It involves adding new genes or DNA, likely from a different species, to make transformational changes in phenotype. It has had a huge uptake in agriculture, where the majority of crops such as soya and cotton are now GMOs. It can be used widely across different traits, with pest resistance and herbicide tolerance the most utilised, but it is increasingly used for consumer quality traits as well. It's an area where Scion has done a lot of work over the last few years, particularly looking at sterility, growth and wood processability. It's one of the key technologies we have used in the Winning Against Wildings programme, and shown here are some transgenic Douglas-fir trees growing in Scion's GMO glasshouse with modifications designed to produce sterile trees. Next, I would like to say a little bit about gene editing. Gene editing allows you to change a gene in a highly specific and targeted way to create a designer mutant with an advantageous phenotype. You don't have to add new DNA, and the changes made are of the same type as occur through chemical mutagenesis, which is a non-regulated process, or indeed through evolution. As I mentioned earlier, this has led to much of the world not regulating this technology as a GMO. So how can we apply gene editing in forestry? The cartoon you can see here outlines some of the examples being used in agriculture. You can see that the applications are very similar to those for transgenic technology, with a focus on stress tolerance and disease resistance. In forestry, yield improvements would come from growth and wood quality traits rather than grain traits as shown here. An important observation is that the first gene-edited crops have now been commercialised, and these are not being regulated as GMOs. From a forestry perspective, gene editing is a really exciting technology with huge potential. For this reason, Scion has put a lot of effort into developing it in conifers over the last couple of years. I'm pleased to say that we've had success with this, although there's still quite a lot of work to do. In this picture you can see some gene-edited radiata trees growing in Scion's GMO containment glasshouse. These have had a cell wall gene edited, and we've looked at the cell wall characteristics and found that they are different. We also have some cell cultures of both Douglas-fir and Radiata with edits in genes we think are essential for reproduction, and so should produce sterile trees. Over the next few months, these will produce seedlings that will be transferred into the glasshouse. The next two technologies I will describe are more recent, and you're probably less familiar with them. These alter the way genes work, but they don't alter the gene's DNA sequence. Because of this, they are not considered to be GMOs. The first of these, exogenous RNAi, uses a cell's own molecular machinery to inactivate genes. The natural role of this process includes things like detecting and destroying non-host RNA, such as from viruses, and regulating gene expression. The diagram shows one of the ways in which this works. In simple terms, a gene, which is encoded by DNA, is copied to produce a messenger molecule called messenger RNA. This messenger RNA is then used as a template to produce a protein, and proteins generally carry out functions within cells, such as enzymes. One of the ways that cells regulate this process is by producing regulating RNAi molecules, here RNAi. These molecules interfere with the messenger RNA to degrade it and ultimately stop the protein being produced and turn off the gene function. There is a lot of knowledge about how this process works, so we can design artificial RNAi molecules to turn off genes. This can be very targeted, as we can design RNAis against individual genes whilst not affecting others. This technology has been widely used as a transgenic tool, but we now know that you can treat a plant with RNAi which has been produced outside the cell. For instance, spray it with a solution. The RNAi then enters the plant and is able to change gene expression. Because this effect only lasts whilst RNAi is present, it's a transient process. The biggest challenge with using this technology is the ability to apply RNAi at the right time and in the right amounts. So far, the main application of exogenous RNAis has been in the development of RNAi-based pesticides, where the target genes are essential genes from insects, fungal or viral pathogens rather than plant ones. These RNAi molecules can be applied to a crop, and attacking pathogens take up the RNAi. This then turns off the target gene, killing or disabling the pest. As you target a particular gene, this is a very specific process and does not affect non-target organisms or indeed the host plant. RNAi also has very low intrinsic toxicity. The RNAis can be formulated with carriers, such as nanoclay, to give slow release and prolong pesticide activity. RNAi can also be used to influence plant gene expression, and I will say a little bit about one possible use for this later in enhancing tissue culture performance. We're also interested to see if we can use the transient nature of exogenous RNAi to turn off genes that repress cone development in Douglas-fir to allow precocious coning. If we can do this, we can use it to test the effectiveness of the sterility constructs that we've made in the Winning Against Wildings program. The final technology that I want to talk about today that can potentially alter phenotypes without changing the DNA sequence utilizes epigenetic modifications. I won't go into detail about this other than what you can see in this diagram. You're most likely familiar with the concept of DNA being a double helix as shown here. But in the cell, the DNA has a much more complex structure than this. The DNA is further packaged around proteins and condensed to form chromosomes. And the DNA in its packaging can have various chemical modifications. And these modifications affect whether and how well genes work, for instance, turning them on and off in response to environmental challenges. A major difference between epigenetic approaches and the RNAi approach we've just talked about is that epigenetic changes can be very long lasting and can indeed be heritable, passing to the next generation. But unlike DNA sequence changes, they are dynamic, so can be altered in response to environmental factors. That's all very well, but how can we utilise epigenetics in breeding programmes? One way is that as we better understand epigenetic changes, we can use them to predict outcomes and use this knowledge as a tool to select desirable phenotypes. There's actually a lot of work around this in human health, as epigenetic changes that occurred earlier in your life, or indeed in your parents or grandparents, alter your risks to various diseases later in life. We're also starting to understand how we can modify epigenetic profiles to change the way genes work. Here you can see a simple example of how this might work. In nature, epigenetic changes can be primed so that a stimulus can prepare the plant to deal with a stress later on. This makes them better able to respond to a stress. We can achieve the same effects using priming stress, chemicals, or molecular tools to give long-lasting phenotypes to things such as drought tolerance without altering the DNA sequence. For my final couple of minutes, I would like to give an example of how we might use these two non-GMO technologies using improvements in tissue culture to outline this. As you might have heard earlier, tissue culture is an enabling technology for the mass propagation of trees to support clonal forestry. It's also the route we use to propagate biotech trees. One of the key steps in tissue culture is the production of embryos from cell cultures, which are then germinated to produce trees. This is the step you can see on this slide with an example of it working well. Unfortunately, the results of this process are very variable. Some genotypes just don't like doing it, meaning that a lot of lines need to be screened to identify good genotypes, and these may not be the best performers in the field. Another issue is that as the cultures get older, they lose the ability to produce embryos. So what you often see, unfortunately, is more like this. We know that both epigenetic changes and gene expression play important roles in how genotypes perform in tissue culture. This opens up the opportunity for screening to identify and select genotypes that are likely to be performing well in tissue culture, reducing the need to do time-consuming testing. But we can also start to think about using exogenous RNAi and modifying the epigenetic profiles to make increased numbers of genotypes more amenable to tissue culture and clonal propagation, thus widening the group of genotypes where we can carry out this process. Exogenous RNAi is particularly suited to this because it has the transient effect we described earlier. So you can add it to cultures when it's required and encourage them to perform better. And when you've done this, you can stop the treatment and the trees will then grow with no effects from the RNAi you added. And just to finish, I hope that I've been able to show you that several existing and emerging technologies are going to provide some really great tools that will enable transformational changes in forest breeding programs. Finally, thanks for listening today, and I'm happy to answer any questions you might have, either in the discussion session that's going to follow, or indeed through my contact details on the final slide.